Hi, let's discuss about one of the immune response, which is humoral immune response. So what is humoral? Humoral immune response refer to immune response that happen inside the body fluid. So basically, we are talking about the blood inside the body. If the pathogen is located inside your body fluid or in the blood system, it is under humoral immune response. So we're going to look at the different cells and different component that involved in the humoral immune response. The first one is the pathogen, example bacteria. The second one is macrophage, or also known as antigen presenting cell. Antigen presenting cell has class 2 MHC molecule. Class 2 MHC molecule is like a receptor, but it is not a receptor. It looks like receptor located on the surface of the macrophage. The function of class 2 is to present the antigen fragment on the surface of the macrophage. Next, we have helper T cells. And then, helper T cell also has its own receptor. Next is B cell. B cell also has its own antigen receptor and also class 2 MHC molecule to present the antigen fragment. We also have the memory cells and then plasma cells, antibody and antigen fragment, and cytokine. There are two types of cytokine, interleukin 1 and interleukin 2. Cytokine is a molecule made up of protein that released by interleukin 1 is by antigen presenting cell. Interleukin 2 will be secreted by helper T cell. So cytokine is like a signaling molecule that going to help the immune response to happen. These are the process. Step number 1. The pathogen need to enter the body. The pathogen could be viruses, could be bacteria. Enter the body, the foreign molecule, and it's will move around your body fluid or your blood system. Once it encounter the macrophage will engulf the pathogen. Let's see, it's going to engulf the pathogen. This is engulf. It's going to engulf. Basically, engulf means it's going to eat the pathogen. And then the pathogen will be inside the macrophage. After it's engulfed by by phagocytosis, the pathogen will be digest and break down. Basically, we're going to cut the pathogen, and the pathogen going to produce a small fragment that we call antigen fragment. This is antigen fragment. Antigen fragment is the molecule substance that produced after the bacteria has been digested by the macrophage. This antigen fragment need to be presented on the class 2 MHC molecule. It will move out and will be presented on the class 2 MHC molecule like this. So, after the macrophage already show the antigen fragment on the class 2 MHC molecule, now you can call the macrophage, as antigen presenting cell, because it's already present the antigen outside of its body. So that is the beginning of the humoral immune response. Now, the helper T cell, which is the middle cell, going to help the next step. The helper T cell that has its own receptor will bind to the class 2 MHC molecule antigen fragment. So, the helper T cell receptor will bind to the class 2 MHC molecule with display antigen fragment on its surface. It's both of them going to bind. Going to zoom in. So it's going to bind exactly at the antigen fragment. This binding will activate. This binding will activate the macrophage. So once the macrophage is activated, it's going to secrete interleukin 1, which is the cytokine. The APC will secrete the cytokine, or also known as interleukin 1. What does the cytokine do? It's going to activate the helper T cell. Activated helper T cell will undergo proliferation. What is proliferation? Basically, the helper T cell going to multiply itself, becoming large in number, and also it's going to differentiate. This going to produce many helper T cell. While differentiate, the helper T cell will form memory helper T cell. This is not in your notes, but that is what actually happened. Now we have an activated helper T cell. Proliferation will make the helper T cell will increase in number. So now we have many helper T cell around the site of infection. This is just an example. At the same time, this is the B cell. B cell has its own antigen receptor. What's going to happen is the antigen, for example, this is the antigen, will bind to the antigen receptor on the B cell, and then it's going to enter the B cell. The B cell will digest the antigen and produce the antigen fragment. This antigen fragment again will be displayed on the class 2 MHC molecule on the surface of B cell. So actually you can see that what happened at the macrophage is almost the same what happened in the B cell. So now B cell, after it displays the antigen fragment, B cell, also known as antigen presenting cell. Now we have an activated helper T cell. Activate helper T cell will come and bind to the class 2 MHC molecule with antigen fragment on its surface. It's going to come and bind to class 2 MHC molecule. After the binding, this is the helper T cell. After the binding, helper T cell will secrete another cytokine, which also known as interleukin 2, to activate B cell. What happened to the B cell? B cell will undergo again proliferation to increase in number. It undergo multiplication and mitosis to increase in number. So we're going to have a lot of B cell at the site of infection. And also B cell, the activated B cell also will undergo another process that is known as differentiation. What happened is the B cell going to change, grow and reshape its structure to form memory B cell and plasma cell. Memory B cell, and also another one is plasma cell. Later, plasma cell will secrete the antibody that looks like this. So many antibody will be secreted by plasma cell, and the antibody interact with antigen to neutralize. It can be by neutralization, complement system, and also opsonization. Alright, so that's it. In conclusion, if you want to write an essay, you're going to talk about this part first: how the macrophage engulf the bacteria, digest the bacteria, and then display the antigen fragment on the class 2 MHC molecule, and then the helper T cell binding to class 2 MHC molecule with antigen fragment. This will activate the helper T cell this will cause the macrophage to secrete cytokine and activate helper T cell. Activated helper T cell will proliferate and differentiate to produce many helper T cells and also memory helper T cells. At the same time, B cell also will bind to the antigen and digest the antigen to produce antigen fragment. Antigen fragment will be displayed on the class 2 MHC molecule. Another helper T cell again will bind to the class 2 MHC molecule with antigen fragment. Helper T cell secrete cytokine interleukin 2 to activate the B cell to proliferate to produce many B cell and also to differentiate in order to produce memory B cell and also plasma cell. Memory B cell will be very important in secondary immune response. The body will already have the memory of the pathogen that enter your body. Last but not least, plasma cells secrete antibody. That's it. Thank you.